your presentation.
Hello, everyone. My name is Gerard Gatch, and I'll be the moderator for today's webinar on the topic of multifunctionality elements of twin column LPLC systems. This is one in a series of more than 10 webinars on next generation multi-column chromatography for applications in the production of protein therapies. We understand you have a limited amount of time and came here to hear the content, so only a few housekeeping notes and we'll commence with the presentation. Today's seminar is approximately 20 minutes long, leaving plenty of time to answer any questions you may have. This presentation is focused on the unique features of the YMC twin column chromatography platform. While the base system has patented features for the use in continuous capture step of therapeutic proteins, the platform has been proven to provide multiple other critical downstream purification steps, all on the same skid. Today's speaker will present the Swiss Army knife-like multifunctionality of this platform. We won't have the time to fully explore many of the features of this broad-based technology in this webinar. However, we invite you to explore other webinars that cover topics such as control, reporting, case studies, and deeper dives on capture and sequential polishing functionalities. These are 10 to 20 minute presentations by subject matter experts. Today's presentation will cover these topics. A twin column system, we'll go over that very briefly for those who aren't exposed to the base technology and the functionality of both the batch and capture SMB. Talk about the feature of buffer inline dilution, sequential polishing, and then wrap up with some insights as to other control platforms, single-use interface. We'll provide a summary, and then again, we'll have plenty of time at the end for questions. So our presenter today, Wayne Netney, is the Global Sales Director for YMC Process Technology, and he's stepping in for our presenter, Dr. Kathleen Milbachler, who's feeling a bit under the weather today. Wayne is the Director of Sales for the biopharmaceutical downstream process equipment for YMC. Wayne has over 30 years of direct sales and sales management experience in the industry. He holds a bachelor's in microbiology from the University of Michigan. Go Blue. Please welcome Wayne Netney. Wayne?
Go Blue. Hello, Gerard. Thank you. And welcome everyone to this presentation. I'm here to talk to you about the multifunctionality of the Contichrom Twin Capture SMB system. But before we talk about multifunctionality, it makes sense to remind you of this skid's most process intensifying capability. That is the primary reason people pursue this technology in the first place. And that is its ability to perform twin column capture. Well, if you weren't all muted, you might ask, what is twin column capture and why would I want it? Twin column capture is one of many chromatographic processes developed and patented by Chromacon, a Zurich-based company and a 2006 spin-off of ETH Zurich, and as of last year, a YMC company. So how does twin column capture work? Well, let's start with a single column process. We know from experience that in a traditional batch process, the resin in your expensive protein A column will only be about 50 to 75% utilized when the product breakthrough first occurs during a capture step. At or just before this breakthrough, you would typically stop loading, then would wash, elute, and recondition the column. After reconditioning, it is once again ready to load. It isn't unusual to run 3 to 5 cycles of capture to process one harvest. In twin column capture, we split the column into two smaller bed height columns, approximately half the bed height. Now, this following animation will show you how, with these two columns, the process works. You start out as you normally would by feeding the first column, and the flow through of that feed goes to waste. You continue feeding until you are nearly at the point of breakthrough. at which point you connect the two columns. So now taking the flow through from the first column and loading it onto the second column. You continue loading through that first column until the first column is nearly saturated, at which point in time you want to make sure that you wash out any of the unbound protein that is in the piping or in between in the column, and you wash that onto the second column, short wash, at which point in time you disconnect the columns, begin your washing and dilution process on the first column, and continue your feed on the second column. That processing of the first column continues, wash, dilution. All the same time you are feeding on the second column. Continuing to feed on the second column, you execute your cleaning and equilibration on the first column. And by the time that the second column is approaching breakthrough, the first column is ready to receive product. And you reconnect the columns now with the second column taking the lead position. You see the process continues as before. Now you are eluting the second column, and you are continued feeding the first column. And here you are back at the beginning of the cycle. The cycling of loading and processing saturated columns continues in a steady state until you have completed processing your entire batch. So, how does this benefit you? The benefits of this process have been proven many times with many different resin types, molecules at a wide variety of concentrations, and using different methodologies for culture, including fed batch, as well as perfusion. The results are remarkably consistent and include doubling or tripling your productivity. We define productivity as the amount of protein purified divided by the amount of resin used within a given unit of time. This productivity gain can be used to reduce your resin costs by 30 to 50%, or allow for much faster processing times or increasing your capacity within the same footprint. Or you can do a combination of those three things. This process and our first pilot system, which we built in 2016, won the 2017 Aspen Award for innovations in bioprocessing. The results from one very successful implementation have been published in Bioprocessing International and have been presented at several conferences. We will provide a link to that publication after this meeting, or you can simply request a copy from sales@ymcpt.com. So now let's move on to the topic of today's talk, the multifunctionality of this unit. The reality faced by most companies is that no matter how quickly you intend to implement twin column chromatography, you will still be expected to perform many traditional one column batch chromatography steps for existing processes and for new processes developed using a single column approach. You may be asked to do the new process, twin column capture, twin column polish, which we'll talk about in a minute. But you still could get called on to do batch capture, ion exchange, HIC. You might be asked to use pre-packed columns or some combination of pre-packed columns and membranes. You also may be asked to consider buffer dilution and a buffer dilution skid to reduce the large amount of tanks and buffers that are stored in typical production suites. So how can you manage all this? How many systems do you really have room for? How many systems do you want to commission and validate? How many do you want your operators to need to be trained on? Well, fortunately for us, the bench scale unit to implement this process was conceived and designed by our Chromacon colleagues in Switzerland. So it should not come as any surprise that they would build in versatility. One of the iconic products developed in Switzerland, beyond watches and chocolate, of course, is this knife, invented in 1890. The knife you were looking at was probably a World War II era tool, which became known by the US troops returning home as the Swiss Army knife. This term has become a common metaphor in the US and refers to any person or product which has a wide variety of uses. Let's talk about versatility now. Remember your challenge. You must prepare to handle the multi-column processes of the future while continuing to support and manage a variety of single column processes used today. How can you do this? I give you the Contichrom Twin. So we have already built this system. It comes with all of the functionality that we described in the previous slide and can execute any of the steps that you need to execute in a typical manufacturing purification suite. We have already built this in everything from the benchtop unit, which is a 30 milliliters per minute system, all the way up to a full production unit of 20 liters a minute, and everything in between. These systems have not just been conceptualized, but these have been built and are operating today, producing proteins. So let's talk about some of the functionality of this system. The process that I just spent a few slides describing, it has many benefits, and I will not belabor that any further, but this is the main function of this skid. Also, the system is a fully functional batch chromatography unit. You can run existing single column processes such as capture, ion exchange, or HIC on this system. A newer type process is one that if you have a one column process that's already validated, but you're experiencing capacity limitations, you can operate this system in what we will call a twin column alternating batch mode. Some people will call this flip-flop, where you have two columns, both running batch, simply moving the feed instead of stopping it when the breakthrough begins. The system, when you include a third pump, also provides inline buffer dilution. There's a tremendous opportunity to reduce the space required and the logistics of using buffers in their process concentration. With onboard buffer dilution, you can use concentrates which are diluted volumetrically utilizing the state-of-the-art metering pumps we use in our system. You can easily dilute most buffers 2 to 50x with a guaranteed accuracy of 1%, although for most dilutions, that number is probably closer to 0.5%. This feature can be used for any buffer, including your cleaning and storage solution, and is critical when using the system for a polishing step where a gradient is required. The last operational mode is sometimes taken for granted, but can offer tremendous efficiencies by combining two polish steps into a single step, which we call sequential orthogonal twin column polish. We can combine these steps in a variety of ways, including flow through, bind dilute, flow through. Now, both of these can be implemented using a combination of packed bed and membrane absorber. You can also do flow through, bind dilute, and any really combination of ion exchange or HIC chromatography, and of course, you can still do single column polish. Now, this function in and of itself is really a lot to wrap your head around. We have devoted an entire webinar on this particular functionality, which can also be found on the BioWebinar page. To summarize some of the operations that I've just discussed, let's review what we've talked about. The system can perform both of the process intensifying twin column processes of Capture SMB and sequential orthogonal polishing, but it can also perform conventional one column batch steps, whether those are capture or polishing. To all of this, add the capability of saving space and improving the logistics associated with ordering, storing, and transporting large volumes of process concentration buffers. We're not talking about a system that will be available at some point in the future either. These systems have been built and implemented from our bench scale 30 mil system to our largest system with process flow rates of up to 20 liters per minute. This table lists the standard ranges for each of our GMP systems and also provides some approximations of the column size you would use and the amount of protein you could expect to process in a day. Now, these approximations are obviously unique to every situation, but are a good starting point for estimating purposes. This slide lists some of the many standards that we are asked to adhere to, and also lists some of the best-in-class components we use in the systems. I'll let you read for yourself, but this is certainly, from an automation standpoint, CFR 21 Part 11 compliance. We, of course, use Class 6 materials, 316L throughout all wetted surfaces. We comply to ASME BPE design standards. The components we use include the Liwa industry-leading metering pump. We add to that a digital control motor to allow for even higher precision. And we use other world-class components such as E&H, Burkert, Optec, Allen Bradley, to name just a few. The system can also be made to conform to CE standards and was developed in a GAMP5 environment. So there are still many topics related to the system that I didn't really have time to present today. These I will leave in this category of topics for another day, which will, as you might have guessed, be covered in other webinars. These include a big discussion on our automation, first just getting you comfortable with how it's operated, how flexible it is. We'll talk about the fact that we do write the software in Rockwell, but have also written software and incorporated this system into a facility which uses Delta V controls. The information can be pushed to your network or stored on the machine, and there's various other automation topics built into that seminar. We also have been asked in the past, how can we use this stainless steel system in our single-use facility? And we will devote an entire webinar on the interface that we have designed and implemented to allow you to use this system and connect seamlessly with the single-use feed and eluent systems. The last subject, which also carries its own webinar, is AutoMAB dynamic control, which is some adjustments available on this system to allow you to accommodate either degrading capacity of your resin or variations in your feed concentration. So in summary, I've covered several but not all of the features of this unit, including its continuous capture modes, batch, sequential batch polishing, all using the onboard buffer inline dilution capabilities. So with that, I will now turn the presentation over to Gerard for some final words, but I'll be back after he's done to answer a few questions.
The presentation was sponsored by YMC Corporation. You may know YMC as a pioneer in chromatography, producing bench scale and prep scale HPLC and LPLC systems, packing resin for both large molecule and small molecule applications, and analytical and prep scale columns, all since 1980. YMC is a global company with manufacturing and offices in over 10 countries. Next slide. Just as a reminder, there are other aspects of this topic which are covered in a series of webinars available at biowebinar.com. In these, we go into depth on case studies from end users who have used the technology and provided data and information and insights on how they've incorporated it, the automation and the operation, which in and of itself has gotten, again, user feedback that has provided us with the confidence this is a relatively user-friendly and simple to integrate for operators to use and install. We talk about process characterization, viral clearance studies, a lot of other features. So please look into the complete webinar series. And I've got a number of questions here which I'm going to tee up. We've got a couple of minutes, so let's start off with a question around the footprint of the system. You mentioned a lot of functionality. What does this do to the footprint of a system in comparison to others? I'm assuming they mean to having multiple other systems.
Yeah, thanks, Gerard. Well, the system is not much larger than a typical chromatography system. The systems can be as small as 1 meter wide by 2 meters long, and probably chest height, maybe 2 meters high. Some of the more complex systems can extend beyond 2 meters long and 1.5 meters wide, with the same height, maybe slightly higher height. So it's they're not really that much bigger than a standard chromatography system.
Thank you. Can I use columns that I already have, or do I need to have special columns? I think that would go across the board for both the capture step as well as the polishing step that you mentioned.
Yeah, the intent of our systems are really to allow you to incorporate whatever you are using for columns or membranes presently, and just execute a chromatography step using our system seamlessly. So it's really independent, so long as the pressure rating of the columns are appropriate for the processes that are being run, and that you will have the opportunity to qualify the column and enter those parameters into the system as part of the recipe setup.
Okay, thank you very much. Next question. Is there a pressure rating limit for single column versus twin column design?
Well, the pressure rating of these low pressure systems to begin with, the very small ones are 10 bar, some of the larger ones maybe 7.5 bar. So the pressure drop issue is that it can go no more than that. When you use 2 columns in series, you are obviously experiencing the pressure drop of both the beds of resin. But in the case of the capture SMB step, you have to remember that the beds are only 10 centimeters typically in height, 8 to 10 centimeters, as opposed to the 20 centimeter bed height that you would use in a single batch capture step. So we certainly are sensitive to the combined pressures of those 2 columns, but you'll find that the 2 columns in series have no higher pressure drop than the single higher bed height column.
Thank you. This is from someone who obviously has attended a previous webinar on another topic called MCSGP, and the question is, can these systems also run the MCSGP process?
And the short answer to that is no for the GMP scale systems, but yes for some versions of the cube. The cube system, the benchtop units available in both 30 milliliters per minute and 100 milliliters per minute can be operated to include the MCSGP unit operation.
Very good, thank you, Wayne. There's 2 questions that are very similar, so I'm going to try and combine them together for reference to time. One is, where can I see one of these systems, and do you have any references that can be mentioned? So I guess this is around if people curious as to where these might be installed and how
Well,
to
we
use
have
them.
we have a couple demo systems, demo pilot systems, the 100 size pilot systems that are available for rental and for demonstrations. We have built systems for both U.S. customers and European customers. The U.S. customers are pretty far along. They've already been producing clinical material on those systems, and they have 3 in total: a pilot scale unit and 2 larger scale units. And I guess I can say that customers BMS, as they have already been on record and have published that article that I referenced previously. But we built several, and we certainly have software that can be demoed remotely, and or when we are all traveling again, we have pilot units that are available.
Okay. Another question. Can I add on the buffer inline dilution at a later date if I don't order it with the system originally, I guess?
I would say the best advice, anything is possible, but the best advice is no. And the main reason for that is the piping that is required to incorporate that third pump, as well as the programming that becomes integrated as part of the software to allow you to use the functionality of the third pump, really need to be incorporated with the original system. So I would say no to that.
Okay, thank you. And I'm looking at our time. I think we're gonna have time for one more question, and then we'll wrap up. Do you incorporate membrane filters in the system? And that could go, I guess, a question for either filters that are pre-column, column guard filters, or membrane absorbers. So you might want to answer both of those.
Yeah, well, I think I touched upon the membrane absorbers before, and for the system doesn't really know if you have a packed column or a membrane absorber, and you just have to adjust your flow rates and pressures appropriately for the device that you're being connected to. Relative to guard filters, and you might as well include into that category bubble traps. We understand that customers have processes that are already validated, incorporating some kind of a guard filter, and in some cases incorporating some kind of a bubble trap. It is not worth it sometimes to try to change the validation of that. So our system certainly can be, as an option, both sides, the two sides of the pumps can both be fitted with filters. You can either have a single filter in line, or you can have 2 filters in parallel, one of which will be in operation, and should that filter become plugged and hit a delta pressure limit, you can automatically switch over to the other filter. Same goes with bubble traps. We certainly have available as option bubble traps, all of which can be bypassed or become part of the recipe, depending on the configuration and the process that you are trying to replicate.
Perfect. Well, again, thank you so much, Wayne, for the explanations on a broad range of functionality. I think you did a great job in pulling it to kind of an overview together of multifunctionality of this platform. We are bumping up the end of our time. We want to be respectful of everyone's time. If you do have other questions, please send those to info@ymc.com. That's info@ymcpt.com. I think I left the PT out of that the first time, so info@ymcpt.com. Thank you for your questions. Thank you for your participation. In the coming days, you will receive a link, give us about 2 days, and to an archived version of this presentation, and a link to the associated reference documents, some of which Wayne mentioned, some other ones which will expand your knowledge on this topic. Please feel free to share these with your colleagues. And a reminder, this is one in a series of webinars, all of which you can find at our host, biowebinar.com. We'll end our session now. Thank you and have a pleasant day.